go ahead and get started. So what I want to make sure to have time to do today are a couple of things. First, I want to make sure that we spend some time on the idea of hypertonic, hypotonic solutions, water movement across membranes. We'll spend some time talking about transport proteins and then do a couple of practice problems. And then with the feedback that I got from the class over the weekend, I'm going to just make a couple of comments at the end of class today. So we'll try to cut off with about 5 to 6 minutes to spare to spend time on that. Let's start with this. So this is basically a pictorial of the practice problem. Just to orient you, here's in the middle here is our phospholipid bilayer. And then on the outside of the cell we have sodium chloride. It was listed as a 0.1% solution. On the inside, listed as 0.9%. What I want you to do is, oh my gosh. All right, talk with a partner about what's going to happen to a red blood cell under this condition, okay? And I may have provided some feedback on your comments, but try your best with this one. And then we'll go ahead and talk about it. Take 2 minutes. Go. Yeah, perfect. What's the bilayer made of? Phospholipids, and those lipid tails are hydrophobic. Therefore, water's not going to pass through here by simple diffusion. If it does so, it's incredibly rare. Where does water pass through? How does it get across? Who said that, Matt? Nice. All right, aquaporin. So aquaporin is one of the main protein channels. I'll just draw it like this real quick. This is just kind of a cross view. So water is going to be moving in one of these two directions across aquaporin. Which direction? Into the cell or out of the cell? Into the cell, because why? What's more concentrated? So here's where we need to be specific with what we're saying. What's more concentrated? Okay, good. The NaCl. So water is going to be moving into the cell. That cell expands in size. Maybe if left for long enough, it'll actually burst, lyse. So the solution outside of the cell is hypotonic, hypertonic, isotonic relative to inside. What was that, Abby? Hypotonic, yeah. So the constant, or where we have a lower solute concentration, we say that's hypotonic relative to the other solution. And the one thing to point out here on these responses, sometimes there was a switch of which is hypertonic and which is hypotonic. The other thing I want to point out here is that these are comparative terms. Just like saying something is greater than or less than, hypotonic is really saying that the concentration of the solute outside is less than it is inside of the cell. So you need to make sure that when using hypertonic, isotonic, and hypotonic, that you're actually using them as a comparison, saying what are you actually comparing between the two. The way that water actually moves through aquaporin is kind of like a ping pong. So the water being polar will kind of attach itself briefly in hydrogen bonds to different amino acids on the inside of that pore there, allowing it to kind of be pulled into the cell. Okay. Quick side note question. Obviously, this is a pore. This aquaporin is a pore protein, so it really looks more like a cylinder, and that cylinder is kind of wrapped by the phospholipid bilayer. What's true about the amino acids on the outside face of aquaporin? Would they be hydrophilic or hydrophobic? What would you expect? Why do you say that, Sid? Why? That's not a bad thought. It is hydrophobic, but it's not exactly for that reason. Think again, where is this located? Where is the aquaporin located? In the lipid bilayer. And so in order to maintain its three-dimensional shape, it has to have some stabilizing forces on the outside of that pore as well. So since it's basically kind of dissolved in this phospholipid bilayer, to make sure that it stays there, this outside face has to be hydrophobic as well. So in some ways, this is an amphipathic protein. The inside of it is hydrophilic. The outside is hydrophobic. Okay? That's just more kind of a review from things we've talked about before. Okay. Does sodium, and this movement of water, does it require energy? No. With the concentration gradient, right? So we'll come back to that in a little bit. All right? What about sodium and chloride? Will they move? So would sodium and chloride move across this membrane in the opposite direction? Yay, nay, I don't know. Why should I care? Well, I'll tell you why you should care. Because if you give somebody a bag of pure water and saline, or pure water as an IV drip instead of saline, then their red blood cells they die from hyponeutropenia or something like that. Can't remember the exact word. It's an anatomy and physiology thing. Salt, chloride, hydrophilic, hydrophobic properties. What do we? They're charged, so will they pass through the bilayer? Not without some sort of channel or pore to pass through. And typically what we find with molecules like, or atoms like sodium, chloride, what we find is that it can be movement across the membrane, but oftentimes it's unidirectional. So for example, a cell may try to trap in sodium, or sometimes we see pumps like the sodium-potassium pump in eukaryotic cells where, let's see if I can remember this, potassium's coming in, sodium's going out, and that requires some sort of energy for that movement to take place. So unless there's energy applied to the system, the sodium and chloride probably won't move through the membrane. At least they're not going to move through as quickly as water would be able to passively. What about with a gram-positive cell? How is a gram-positive cell going to be affected under the same sort of conditions? So gram-positive cells, will they burst like a red blood cell, or will they be okay? Sarah, do you think that gram-positive cells will burst? Why do you think that? Okay. Okay. So you're saying that gram-positive cells have this single phospholipid bilayer. What else do gram-positives have? True, teichoic acid. And they got a thick layer of what? What's that? Or cell wall, right? And that cell wall is on the outside of this membrane. And that cell wall, I'll just kind of, this is a really bad rendition of it, but those are the amino acid bridges that kind of hold the individual strands together. In this conformation here, imagine this in 3 dimensions, this is kind of a cross section. As water rushes in, that cell will expand, that membrane will increase in size, right? But at some point, what happens? It hits the cell wall, and the cell wall actually prevents it from expanding even further. Has anybody had celery lately? Cindy, how do you store celery at home? Yeah, like if you're putting it in the fridge. This is a serious question. It's important. Do you put it in water? Oh,
actually, my parents do.
Okay, why would you put it in water? Why would you put celery in water? So it doesn't dry out, and it stays nice and crisp, right? Why does it stay crisp? Because it's constantly it's constantly under water pressure, right? So if you put it in pure water, for example, what's going to happen is water is going to fill up that cell, and then you're going to start to get that membrane pushing against the cell wall of a celery cell, okay? And as it's pushing against that cell wall, that's turgor pressure, okay? That's why it stays nice and crisp, unlike limp, disgusting celery. Nobody likes limp celery. So you've got this turgor pressure pushing on the outside of the cell. It kind of balloons up. But that peptidoglycan, given that it's so strong, prevents that membrane from expanding even further. Okay, it's kind of like a cage or a netting around this to prevent that cell or that membrane from bursting. So this is why, for example, some of those gram-positive organisms can remain in high water or deionized water for long periods of time and actually survive that condition. Now granted, over time they're going to die because there's lack of nutrients, for example. Or that peptidoglycan starts to break down, and once that happens, then the cells will actually burst. But initially, there's a prevention of breakdown there. Okay. Okay, so any questions on hypertonic, hypotonic, isotonic, gram-positive cells? We'll come back to gram-negative a little bit later. All right, let's move ahead. What's the difference between active and passive transport? Or what are some differences? I heard one. ATP is a big one, right? ATP is a big difference between the two. So is ATP required for active or passive? Okay. So I'm just going to set up a quick table here. Active, passive. So active, we say, I'm going to actually define this a tad more broadly and say that it requires energy. And ATP is a great example of that. What else? What's another difference between these two?
If it's active transport, it's going like against the concentration.
Good. Usually pushing against the concentration gradient. And how about passive? Just the opposite, with gradient, right? These are kind of, when we compare the two, these are kind of the biggest differences between active and passive. You may also see the word facilitated, and facilitated is a lot like passive. There are just minor nuance differences. So in passive, the channel itself remains unchanged throughout as the molecule moves through the channel. In facilitated, we see some minor changes in shape to the actual transport protein itself. A prime example is the glucose transporter. It kind of opens up on one end, the glucose comes in, it changes shape and allows the glucose to enter into the cell. And that's a passive, or that's a facilitated transport mechanism. However, we actually don't see glucose exiting these cells after it's gone in, even though it's facilitated transport. Why don't we see, for example, glucose moving back out after it's entered the cell? Anybody know? And it actually comes down to the glucose gets modified. Glycolysis lovers. Matt, you're a glycolysis lover, I'm going to assume, because you're chemistry, right?
Yeah.
Yeah, okay. What's the first step of glycolysis? Oh, damn it. Anybody remember the first step of glycolysis? Glucose gets turned into something. Gets phosphorylated. Pyruvate's all the way at the end, Zach. That's 10 steps later. Now that's even further. Now you're talking Krebs cycle. Yes, I'm positive. I'm not going to force you to memorize, but I'll tell you I remember like 5 steps, and those are 2 of them. First step, glucose gets phosphorylated into glucose 6-phosphate by the enzyme hexokinase. Okay, and there's other ways to do it as well. But that modification right there, adding a phosphate changes the structure of glucose. By changing the structure, that glucose molecule is no longer, we could just say that it's no longer recognized by the glucose transporter, and so it can't actually exit the cell. It essentially gets trapped inside. And we see this actually quite often with bacterial cells, that they have transport mechanisms that ensure a unidirectional movement of nutrients and other trace chemicals. So a classic example, at least from the perspective of the human body and pathogenicity, iron is a limiting factor, particularly iron in its Fe2 plus or Fe3 plus form. What we find is that, at least in pathogenicity, we describe as there being a constant battle with bacteria. That since iron is a trace nutrient, our cells are trying to grab it, bacterial cells are trying to grab it. Bacterial cells release what's known as essentially siderophores. These are iron scavenging molecules. They secrete them from the bacterial cells, and at random, these scavengers will reattach to the bacterial cell, and a transport protein using active transport then brings these iron molecules in. And by using active transport to bring them in, it essentially locks that iron now into the cell, therefore ensuring that those iron molecules are moving against the concentration gradient so that there's more of it inside than outside of the cell, and it's going to help these organisms to perform metabolic activities. So oftentimes when we see active transport, I'm just going to pull up an example a couple slides down here. Here are 3 of them. When we see these mechanisms of active transport, they're often because these molecules are in trace amounts outside in the environment, and so bacteria are trying to bring them in as a way to encourage or support growth. Am I expecting you to know each of these examples? No. What I want you to be familiar with are general themes, and these kind of hit on some general themes. So one is each of these represents a use of energy to allow a molecule to come in. This one, hydrogen ions are going with their concentration gradient, and that energy that's generated is coupled with another molecule going against its concentration gradient to bring it into the cell. Here's an example of a glucose transporter where phosphoenol pyruvate, right? Okay, it did come into play today. High energy phosphate on here gets transferred to the glucose molecule, thereby locking it into the cell. And then we've got kind of our classical example here where addition of ATP or using ATP to change the shape of the transporter in a way to help facilitate the bringing in of a molecule here. And those are just known as ABC transporters. I don't need you to know the specific examples. Really what I'm just trying to hit on are the generalized themes. Against concentration gradient and requiring some form of energy here. Okay. Let's see if there's anything I'm missing from this that I wanted to make sure to get to. Nope, I think that's about it with those. Okay, let's take a look at an example. All right, so here is a membrane. Here are some proteins. Does this look familiar to anyone yet? Jenna, what is this? Yeah, it is. This is the electron transport chain. All right. So we've got, in this case, H+, sorry. Okay. in this example here, I want you to tell me what kind of transport is occurring at letter A and letter B. Okay. I'll give you a few minutes, probably like 3 minutes to think about this, but what kind of transport is occurring? Let me add one more piece here. Okay. And what I want you to do is not only say what type of transport, but what's supporting that argument. Okay, go ahead. So for this one, there's a couple of important pieces of information up here, right? And there's a reason why I added pH on here. Otherwise, you may not necessarily be able to figure out. But I think the pH kind of helps. So pH is a measurement of hydrogen ions. And as pH decreases, increase in hydrogen ion concentration. Roughly, the equivalent is for every drop of 1 in pH, so if you were to go from 7 to 6, that's a 10-fold increase in hydrogen ion concentration. So the difference between 7 and 5.5 is really like a 30-fold difference across the membrane. And so in A there, hydrogen ions are going in what direction? With or against the concentration gradient? Going against. Therefore, this must be what kind? Active. Where's the energy coming from?
It uses ATP to do that.
Ah, does it?
Or ATP training,
Does it? No. Where's that energy coming from?
would it be that hydrogen?
No, it's not the hydrogen ions itself. Somebody said it. It's the electrons. It's the high energy electrons. So we'll come back to this in Wednesday, Friday, next week, somewhere in there. But those electrons we consider to be high energy as they're coming off of coenzymes, NADH, for example. So as they come off there, and as they move down that chain from one to the next, they lose energy with each step. And as they do, that energy that's released is used to help move those hydrogen ions across. Okay? This step here, active or passive? Passive I would go with because it's going with the concentration gradient. There's no energy to move it in. It's not that simple transport where I showed the hydrogen and another molecule moving in at the same time, because it's not actually transporting anything else across. The ADP and the ATP, or sorry, ADP is already on the inside here, and as this rotor moves, that helps to create the bond between the ADP and an organic phosphate to produce ATP. So it's kind of energy capture from here, kind of like a waterfall to allow for production of ATP. I was going to say one more thing. Oh, what other system is like this? Anybody recall we talked about another system that was kind of like this, where hydrogen ion passive transport drives something. Maybe rotate something required for motility. Flagella. Flagella has a similar mechanism, not exactly the same, but some similarities in there. Okay? All right. How we doing? Any further explanation? So, Sarah, go ahead. No. No, I will take active transport as an answer, and that's perfect. So again, the key things you're looking for are what's the energy source with or against the concentration gradient are kind of your key themes to be looking for there. And that's what I would be looking for in an answer with this. So I've never used this one on an exam, but used it in review actually quite frequently because I think it's a really nice example because it takes us away from like classical examples and moves us into something that we will come back and see again a little bit later. Okay. All right. Shall I try one more problem for the day and then we'll talk a little bit? All right. We'll go with chalkboard black this time. All right, you've got a gram-positive bacillus, gram-negative cocci, and let's say that, I need a different color. Yellow. Blue. Okay. So let's say you have two beakers. One with pure water, distilled H2O, and another one with pure water, but you've also got penicillin. The gram-negative survives well in the water for like 5 minutes. It also survives well when the penicillin is there as well. However, the gram-negative explodes. Or, sorry, gram-positive explodes. Huh, why? Okay, so why does that happen? Have we seen this problem yet? No? Yes? Who's going, what the hell did he just do? Why ask this question? Take a shot at it, okay? Use what we've talked about not only today, but Friday, probably Wednesday as well, to help guide you with this one. And then we'll talk about how to approach it. Or you use it.
What's in the gram?
Okay, so compare and contrast gram-positive and gram-negative. And actually, this question is designed to force you to do that. Exactly. So what are some differences? Two cell membranes on gram-negatives, an inner membrane and outer membrane, and gram-negatives gram-positives just have one. What else is different? Gram-positives have thicker peptidoglycan than do gram-negatives. And if a cell is going to burst, what's that a result of? What's likely a cause for that? Too much turgor pressure. But didn't we just say a while ago that gram-positives don't burst because they've got this really thick cell wall that prevents them from bursting? So what's that indicate? Broken down. Right? So here's where you can start to pull pieces from multiple lectures together to get the answer. We've got cell wall must be important. We've got one difference is the outer membrane. Why don't gram-negatives burst? Because that outer membrane is semi-permeable, possibly. And in fact, that's actually what happens. That outer membrane is impermeable to penicillin. And so what we found early on with the use of penicillin works really well against gram-positives. Limited use against gram-negatives. Okay? That penicillin, it unlike lysozyme, which we said actually hydrolyzes and breaks down those between in those polysaccharide walls between the NAG and NAM. What penicillin does is it actually prevents new peptidoglycan from being formed. And this is something we'll cover probably 8 weeks from now. We'll talk about the exact details of it. But the preventing new wall from being formed, what that means is as this wall begins to deteriorate, it starts to weaken, and over time, these cells will continue to expand and grow, but that's going to also lead to the weakening of the wall, and they just kind of blow up like a popped balloon, essentially. Okay. Do you need to know about penicillin to answer the question? No. And in fact, I wouldn't necessarily ask you what penicillin does. This is the kind of style of question that I will put on the exam. And what I mean by that is it's not something you've seen, but you've seen something like it. Or there are, if you can pick out some fine points like, yeah, gram positives versus gram negatives, difference in outer membrane, difference in peptidoglycan, those all get you points on those types of questions. My essay questions are not all or nothing questions. There's a lot of partial credit. As myself and friends in organic chemistry used to say, we live and die by partial credit, and that is true. Okay. So for if this were to be an essay question on an exam, what I'd expect to be seeing are conversations on the differences of gram positive and gram negative, maybe some discussion on turgor pressure or the movement of water into the cell, weakening of the cell wall. If you don't have the answer correct based on evidence, that's fine. I don't expect you to. I'm looking for consistency, so a consistent story throughout, and also that you are using factually correct information, or at least things that in theory are factually correct based on what we've talked about in class. Okay? Whew. All right, good timing. Any questions on this one? All right. So what I want to do is I want to just take a few minutes and cover some things that were said on not only from this class, but also from the morning class in terms of the setup of class, and then I'll just give a couple of comments too. The first one is, I think people in general like the resources that are available. It's not for everybody. I completely understand that. What I'm trying to do in here, at least, is to provide a couple of different ways for you to get information. One, me just going on and on. Two, you talking. And what I'm seeing in these groups is even like somebody will explain the answer to me, a partner may not get it, and then I say, yep, you're correct, and then they explain it to the partner. That right there, teaching somebody else, I find is one of the best ways to learn information. Because soon when they start to ask questions, you're like, I don't know how to answer that. You figure out where your own faults are. Okay? So continue to do that. And then if you are somebody that wants to work privately, by all means, take the time to do that. So we're trying to build each of those levels in here throughout class time. Every single semester, never fails. I can already tell you what my student opinion evals are going to look like. The worst mark is going to be organization, because it is every damn semester, and I can never seem to get a hold of it. What I have realized about myself is I no longer think very well linearly. I think I might have some ADD tendencies. And what I mean by that is, number one, know yourself, right? Know thyself. What I mean by that is when I think about things like the cell membrane, immediately my thoughts go into 3 different directions or 4 different directions as to what could be going on. Just this problem right here, we already said gram positive versus gram negative. What are the differences? What could actually contribute to this? How does tonicity play in? So what I'm trying to say is that the information is interconnected. It's designed to be like that. And what I'm trying to get you to do is to think a little bit more globally. If you need to rein me in and say, whoa, too fast, too soon, slow me down. Okay? Ask questions to get me to kind of walk through it a little bit more so that maybe, if you're not frightened, you can see into the thought process. Okay? So those were a couple of things. Oh, I have one small concern. And I'm not sure if this is just the way that I had asked the question or if this indicates something a little bit more in depth. A lot of people were saying, I'm keeping up with the lectures, I'm keeping up with the homework. I only heard a couple of people say, and I'm doing review questions. And maybe the question wasn't phrased that way. But what I'd urge you to start doing is spend some time with those review questions. Those are really going to tell you, do you actually understand this well enough to be able to explain it on your own? And I would suggest talk out the answers with somebody else, talk to yourself, which can look good, or write out the answers, okay? And keep practicing with them. And that's going to give you a good idea as to how are you doing as you approach the exam? I don't view these review questions as a only do when the exam is coming, but trying to keep up with them. Do a couple every other day as a way to just make sure that you're applying that information. Okay? Now, let's see. Okay, so couple of things that I also noticed that I wanted to make comments on. One was some folks want more application questions, more review questions. We did a couple today. And I'm not sure if it's really noticeable, but we've actually done some in every single class, at least one. So you are getting that. I will try to pull in more, but it's also dependent on where is everybody coming into class. Like today, we needed to spend some time reviewing tonicity, so that can take away from some of that. If I'm going too fast, slow me down, which I said. And then 2 more here. Sid asked me early on, I'm just going to point you out real quick, Sid, this is nothing bad. Sid asked me last week about, like, I'm reading but it has nothing to do with what you're lecturing in class on, and then I'm doing the mini lectures too.
Well, that's not what I meant. I just said like go reading and go as in depth.
Oh, gotcha. Okay, well that too. Here's what I would say. If you're a visual or audio learner, go all the way back to the beginning of today. Here's what I post at the beginning of class. Evaluate how structure of phospholipids and membrane proteins affect the movement of molecules across the cell membrane of prokaryotes and eukaryotes. We did that in every single example today. So if I'm doing my job and if we're doing in class what we should be doing, those major points should align exactly with these objectives up here. Okay? And that's where the focus should be, is trying to reach these. Okay. Anything else anybody wants to add? All right. Other than that, I guess that's all I've got today. Did John mention to the earlier lab about the assignment that's due in lab on Friday? Okay, small one. This next homework assignment is a VoiceThread one, so make sure you can get access, and it's making a dichotomous key. Very simple one, like on candy. How do you choose from 6 different candies based on the qualities that you want to have in a candy? Blah, blah, blah.
Is it due tonight?
No, it's due, I think I have it due at like 1 tomorrow, only because I wanted you to provide feedback on somebody else's. What was due tonight, thank you for asking that, was the blue portion of the journal entries. Don't worry about it. We covered it in class today, so if you've done it, I've given you the points for it already. Okay? So no need to go back to those journal entries. Move on to the dichotomous key homework. Yes, you'll still get credit. Absolutely. All right. Have a great afternoon, everyone.